Hi everybody, so today we're going to be talking about astrocytes. And what are astrocytes? Well, basically astrocytes are a type of what we call a glial cell. Glial cells are basically cells, glial means glue, right? So glial cells are basically cells in the nervous system that are not nerves, and they make up about half of the nervous system. Now as you can see here, it has the word astro, and that's because astrocytes are star-shaped. Of all the glial cells, this is probably the one with the most function. So let's go ahead and jump into this, and let's talk about why they call this an astrocyte. I'm going to draw my astrocyte right now. By the way, and cyte means cell, right? So we're just going to draw the body, and we're going to call this the soma. And then coming out from the soma, we are going to have what we call processes, right? And you have a bunch of these, and one astrocyte can actually be in contact with thousands and thousands of neurons, okay? And so when they looked at this under a microscope, they said, hey, doesn't that look like a star? Yeah, so let's call it an astrocyte. So at the end of these processes, right, so we have these processes. And at the end of these, what's going to happen, you're going to see these are going to play a big role in just a minute. And at the end of these, what you're going to have is what we call end feet. Okay, now I'm not going to draw it on all of these, but basically at the end of these, you're going to have what we call end feet. Okay, so that's basically what these look like. They're star-shaped cells that are in the nervous system. Again, you have end feet going all the way around here, and the fact that you have all these processes that can actually go on and connect to thousands of neurons. So let's go ahead and take a look at the function of these. So if we were to look at the functions of astrocytes, okay, the first one is going to be they provide support. And I'm going to go through all these in just a minute. So the first thing is they provide support. The second thing they're going to do is they're going to be responsible for injury repair to the nervous system. The third thing they're going to do is they're going to play a role in the blood-brain barrier. And then the fourth thing they're going to do is they're going to help maintain homeostasis. Now, when I say they play a role in the blood-brain barrier, what they can do is they can actually provide nutrition and things such as that and determine what can come across. But we're going to go into all that, and there's a lot down on these last two. So let's go ahead and get started. Oh, wait, before we do get started, well, no, let's go ahead and get started. There's two types of astrocytes. The first type, there's actually three. The first one is called a fibrous astrocyte. Fibrous astrocytes are usually found in white matter. The second type we call protoplasmic astrocytes. Okay, protoplasmic astrocytes, these are going to be found in gray matter. The protoplasmic astrocytes actually contain a protein that we call glial fibrillary acidic protein, or GFAP for short. Okay, so that's going to be in the protoplasmic astrocytes. The third type we're going to talk about is basically what we call a radial glial cell. Okay, and basically these are not astrocytes yet. This is going to help with the formation, and that's where we're going to start when we talk about support. So let's go ahead and get started on here. And basically what my radial glial cells are going to do is when the nervous system is forming, they are going to basically come in. Now the nerves will have already started being formed, and what my radial glial cells, I'm just going to call them RGCs for short, what these radial glial cells will do is they will actually guide the astrocyte and the axon, not the axon, the neuron. They're actually going to guide the neuron and its axon on its placement in the nervous system. So I have my radial glial cells going all through here. Okay, and this is my neuron here. So it's going to guide it and basically show it where it's supposed to be and provide scaffolding, and that's what we call this, scaffolding. okay, because it acts like a scaffold, right? Just like you see on a building, that's what these are going to do, and they're going to guide these into place. Then eventually these radioglial cells will turn into astrocytes. Okay, so they can also turn into oligodendrocytes, but for the most part, they're going to turn into astrocytes. Okay, so that's the first thing it does. Now, when everything's real mature, what's going to happen is these astrocytes, those radioglial cells will then turn into astrocytes, like we just said. That's the support it provides. Now, let's take a look at the second thing. We talked about injury repair. Okay, so let's go ahead now, and I'm going to draw. This is the cerebrum, the big part of the brain. This is the cerebellum. The cerebellum hangs like a bell off the back of the brain. This is on a person. You're looking this way. You're looking at this from this angle on someone facing that way. So if I were to draw the eyes, this person would be looking that way. Let's say they get some type of damage in here, right? They have a blow to the head, and they get some type of damage in here, and then this area gets injured. Well, what my astrocytes will do is they will start to multiply. And their little processes that we talked about, what they will do is they will hypertrophy and they will become bigger. And then what they will do is they will actually start to cover this up, just like scar tissue covers up cuts when you get them on your skin. We call this, on the injury repair, we call this glial scarring. Okay, so my astrocytes are going to multiply, and then this GFAP, that's also going to be expressed also. Now, at one time they thought what happens is once this is done covering that up and we have the glial scar there, that was it. But now they're starting to find out that this will actually release growth factors to try to repair the neurons that are in that area. So that's the second thing that this is going to do, is it's going to be responsible for glial scarring. Let's take a look at a third function. So we said it plays a role in the blood-brain barrier. So I'm going to draw a regular capillary that's outside of the nervous system. And these are going to be the cells, and we call these endothelial cells. Now in real life, these are flat. I know I'm making mine look like a box. All right, and then there's one portion, again, we're looking at a cross section here, okay? So here's another portion. Okay, so these are endothelial cells here. So now, normally this would be the lumen here, right? This is where my blood's gonna be, right? This is normally where I have blood. So what's gonna happen now is normally when a fluid needs to go from here into or leave the bloodstream, it usually goes between these endothelial cells, right? And then it can go into this interstitial space or extracellular space, right? That's what usually happens with fluids and things that are needed. This is when it's outside of the nervous system. What's gonna happen now is let's use purple. You have those end feet. Remember we talked about the end foot? So here's what's going to happen is in the blood-brain barrier, like we said, this is going to be this is an end foot because I'm only drawing one. Okay, what's going to happen now is what the astrocyte will do. Again, this is the astrocyte, this is the end foot. What it's going to do is instead of having these little gaps that are here, it is actually going to get proteins, and it's going to put proteins in here, and it's going to form what we call tight junctions. So when you have these proteins and you form this tight junction now, it's harder for things to get through here, and now this membrane has become more selectively permeable. So what my astrocytes will do in the blood-brain barrier is it forms tight junctions. And therefore it's harder for things to pass through the blood-brain barrier. Now some things can pass through, right? Some things that are lipid soluble or some ions that can pass through also. Some drugs can pass through. So basically that's what it's going to do in the blood-brain barrier. But there's also more. So let's take a look at this. Let's say that we get a neuron and its activity has increased, right? It's starting to fire and it needs nutrients. So what's gonna happen is, let's say we have glucose in here. So here's my glucose molecule. I'm gonna draw it just like this. There's my glucose molecule. So now, what can happen is on these cells, you have a protein. And this protein, we're gonna call, you can pronounce it glut, or you can pronounce it GLUT. We're gonna pronounce this GLUT1. I have a GLUT1 there. I'm gonna have a GLUT1 here. And then we're gonna have a GLUT1 over here. Now, what this stands for is glucose transporter 1. That's a glucose transporter 1. So now what's going to happen is my glucose is going to go through here and it's going to come down into this end foot. When it gets into the end foot, what this glucose is going to do is, or what can happen is, once it's in here, is a few different things. The glucose can be stored as glycogen until it's needed, right? The other thing that can happen is this glucose can be broken down into 2-pyruvate. And then the pyruvate is then converted into 2-lactate. Okay, that's going to be converted to 2-lactate. Now what's going to happen is these lactate can leave this astrocyte through a protein that we're going to call MCT1. And then it can come out through here, and it's going to go into this extracellular space. And then what's going to happen is it can go, let's say there's a neuron over here. So now I'm going to draw my neuron. Okay, so now I have a neuron, and what can happen now is on this neuron, we're going to have another protein called MCT2, and now this lactate can go into the neuron. When the lactate goes into the neuron, what's going to happen is we're going to have the lactate be converted into 2-pyruvate. So it's the exact opposite of what we just saw, right? Because here we go from 2-pyruvate to 2-lactate. Here we go from 2-lactate to 2-pyruvate, which then is going to be converted into ATP, right? That's one thing it can do. So now my astrocyte can actually store this as glycogen, or it can take it can break down the glucose through glycolysis into pyruvate, then into lactate, which can then go through all this and become ATP. But the other thing that can happen too is also over in here, what can happen is you can actually have glucose come through one of these, and then instead of going into here, it can actually go right into the neuron. So let me write down that this is your neuron. Now what's going to happen is you actually have a transporter called GLUT, or if you want to call it GLUT, you can, but this is 3. Okay? And then it's going to come through here, and as you know, now we have this glucose in here, right? We have this glucose, and now this glucose can be broken down, and we're going to go through this process and make ATP. So this is the direct route, this is an indirect route, but either way it can work that way, and that's the role that the astrocyte's going to play in there. But the astrocyte's going to play another role in here. What can happen is water, it's hard for water to get through here, so the astrocyte can actually regulate the water. On the astrocyte, on the end foot, you actually have a lot of proteins that we're going to call AQP4, aqueous pore 4. And what's going to happen now is when you have water on here, the water is going to come through, and it's going to come in, and it's going to go to this aqueous pore 4, and then it can enter into the end foot of this astrocyte, right? Well, what's going to happen now is, and it carries out its normal functions, right? But what's really cool is outside of this, in the nervous system, you have a fluid, or you have something called brain extracellular fluid. And this brain extracellular fluid, what it likes to do is it likes to combine with cerebral spinal fluid, right? It likes to combine with cerebral spinal fluid. So at night what happens is the brain extracellular fluid can't get to all parts of the brain if I have a big old astrocyte there, right? And then remember, we have a whole bunch of astrocytes here and a whole bunch of end feet. So it takes up a lot of space. So for this brain extracellular fluid to get around, it kind of has to go between everything. Like you can see over here, it has to go between this area here. What's really cool is at night, this aqueous pore 4 actually closes off at night and water stops coming in. And because the water stops coming in, these actually shrink at night. And when they shrink at night, it allows this brain extracellular fluid to get to more areas in the brain so it can bathe those areas in the brain and then take waste and things like that and bring it to the cerebrospinal fluid or mix with the cerebrospinal fluid and then go into the venous system and be flushed out of the body. So that's pretty cool, okay? So that is the role of astrocytes on the blood-brain barrier. Okay, that's the role of astrocytes on the blood-brain barrier. Let's look at how it's going to maintain homeostasis. And there's a few ways it's going to do this also. Now, let's draw an axon. And then I'm going to draw a synaptic terminal. Right? So this is going to be my presynaptic terminal. This is the postsynaptic terminal. Okay? And then down in here, I am going to draw another end foot. Okay, remember, I'm writing end foot just because it's one, but you would actually have a whole bunch. So most of the time when we draw synapses, what we do is we just draw these parts, right? We just draw these parts. Now, here's the role that astrocytes are going to play in this. Let me just put right here. This is my axon here. Okay, there's my axon. So here's the role this is going to play here. You have vesicles in here, presynaptic vesicles. Inside these presynaptic vesicles, in this case, you are going to have a neurotransmitter called glutamate. Okay, so there's glutamate. Glutamate is an excitatory transmitter, so when it's released, it's going to cause things to be excited. So here comes my impulse coming down here. My impulse comes down here, and then what's going to happen is I am going to have this glutamate be released. So here comes my glutamate, and it's going to go across the synapse, and it's going to excite the opposite side of this, and then my impulse is going to continue on or do whatever function it's going to do on that side, right? Okay, so now, here's what's going to happen is now we have to collect this glutamate because we don't want this glutamate going and constantly exciting this side. So now it's going to be released, and it wants to go back into here, right? And it comes back into vesicles. So there's a few things that can happen here. The first thing that's going to happen is you have a protein here, and this protein is going to be called EAAT3. And what's going to happen with that EAAT3 is I am going to have 2 sodium come in. I'm also going to have a hydrogen come in. There's my hydrogen. And when those come in, what's going to happen is my glutamate is also going to enter back in here, and then it can be put in a vesicle. Now, as those are coming in, I'm also going to have a potassium going out. Okay, so that's my EAAT3. Here's the thing about this. This, though, can get overloaded because so much glutamate's been released. So what are we going to do? The glutamate's got to go somewhere, because believe it or not, if glutamate's out too much, it could actually hurt the axon, or the neuron. So anyways, what's going to happen is this. Down here on the end foot, I also have other proteins. And these proteins are going to be called EAA1 and EAA2. AAT2. Now, here's what's going to happen, is this excess glutamate is going to come down and it can enter into here. But how does it do it? Well, guess what? We can have, by the way, you can call it either one of these, EAAT1, and I'm not distinguishing them, but just so you know, it's not EAAT1/2. It's there's two different types of proteins. So here's what's gonna happen, is I'm gonna have 2 sodium, a hydrogen, and a glutamate molecule, and they're all going to enter into here. Okay, they're all going to enter into there. And then once again, what's going to happen is I'm going to have my potassium, and my potassium's going to leave. Okay, but now the glutamate, instead of sitting around in here and causing extra excitatory impulses, it's going to come into this end foot. So here's what's going to happen when it comes into the end foot. Okay, we are going to have my glutamate. Here's the glutamate. Okay, there's the glutamate. And what's going to happen is the glutamate is going to mix, or it's going to be acted upon by something called glutamine synthase. And what's going to happen when it's acted upon glutamine synthase, this is going to be converted into glutamine. Okay, so this is still all inside the end foot here. Right? And then what's going to happen is I'm going to have a protein here, which we're going to call SNAT3, and there's another one called SNAT5. Okay, and I only drew one, so if you want to call that 3 or 5, I don't care, but you have 2 different types, right? Just like over here, we had 2 different types of EAATs. So what's going to happen now is this glutamine is going to come out into this brain extracellular fluid. And then on the other side of this, we're going to have another protein, and this is going to be our SNAT1 and 2. Okay, so basically we have SNAT3 and 5, and then we have SNAT1 and 2. Okay, so the glutamine is going to come out. It's going to go through SNAT3 or SNAT5. Then it's going to come in and go through SNAT1 or SNAT2, but it's going to come back inside here. And just for what it's worth, I'm not going to draw them, but you're going to actually have a sodium come out and a hydrogen go in, and then a sodium is going to go in with the glutamine. So if the glutamine comes out, so does sodium, and hydrogen goes in. Here, just sodium going in. So now, I'm going to get my glutamine, and it's going to enter back into this presynaptic terminal. Okay, and then what's going to happen is an enzyme called glutaminase is going to convert this into glutamate. And then what's going to happen is that's going to go back and enter into a vesicle so it can be reused. Now, that's an excitatory. Sometimes what we need is inhibitory neurotransmitters. So what we're going to do is we're going to make GABA, right? So what's going to happen is this can actually come and an enzyme called glutamic acid decarboxylase can actually convert this into GABA, which is inhibitory, right? So that's if you want something that's going to be inhibitory. So that's basically one of the roles that this plays when it comes to synapses, okay? Let's take another look of what this can do. Now, there's 2 more ways that the end feet can help maintain homeostasis. So I'm going to draw another axon, and here's my presynaptic terminal, right? Presynaptic, and then over here I'm going to have my postsynaptic terminal, right? And then over here we're going to have the axon. So here's what happens. 2 things are going to happen. One, when it's at rest, what happens is we have sodium leaking in, and we have potassium leaking out, right? Potassium comes out, right? So here's what's going to happen. I'm going to draw this down here because it'll make more sense. Here's what's going to happen. Once again, I'm going to have my end foot here. So let me draw my astrocyte here. And this is going to be the end foot. Okay? So here's my astrocyte. So now what's going to happen is as this potassium comes out, it should go back in. Now, the polar membrane of an astrocyte has a voltage of about negative 85 degrees millivolts. And as you know, the axon membrane is about negative, depending on the book, about negative 65 millivolts. So you can see this is actually more negative than this. So because of that, an astrocyte actually has a bigger affinity or attraction to these positive potassium. So here's what's going to happen, is potassium is going to come through, and it can come through channels, pumps, gates. It's going to come into the inside here, right? As we get excess, we're going to start to get that in there, okay? And then what's going to happen is as it builds up in there, the astrocytes, I'm just going to draw an astrocyte here, the astrocyte can actually form a gap junction with another astrocyte, or it could be an oligodendrocyte, and this is going to be connected by a protein called connexin. All right, and now we got our gap junction, right? And then here's another astrocyte, and then I'm going to form another gap junction. I could do this several times, or it doesn't really matter how many times I do it, but just to give you an idea. And then here's another astrocyte, and what's going to happen is as this potassium comes in and builds up, the astrocyte can actually have it go into other areas as it comes in. Okay, and then it can kind of store it. So it's going to maintain homeostasis by basically kind of storing the amount of potassium that's in here. And then eventually when this potassium is needed, it can be leaked out for where it's needed, okay? So that's just in the normal function of the cell, of the neuron as it's just at rest and things such as that. But it can also happen when it fires. So let's take a look at this real quick of what's going to happen. As we know, when a cell fires, right, or depolarizes, we get a whole bunch of sodium going in. And then after this gets up to about plus 30, what's going to happen is we're going to get a whole bunch of potassium rushing out, right? Repolarization, right? Well, as we get this buildup of potassium here, what's going to happen is, we've already mentioned how potassium go in here, it's going to change the polarity of this membrane. So when it changes the polarity of the membrane, and this becomes more positive, just a little bit, we're going to have gates that open. And the potassium is going to basically, it's going to come in, but what's going to happen is when that changes, and we have this potassium out here, the potassium is going to start to turn glucose into the lactate, like we mentioned earlier, right? Because it's sensing that this needs energy, so it's going to start converting that glucose into lactate. In fact, the glycogen to glucose to lactate, right? So now that's one thing that's going to happen. But here's the other thing that's going to happen. As that changes, as that polarity changes, you're going to have gates that open on here. And what these gates are going to do is they are going to allow sodium to come in. So now here comes my sodium. And now sodium comes in. But the other thing that's going to come in is something called bicarbonate. I got HCO3 is going to come in. And we're going to get about 2 of those coming in. Now, if you remember, this is a base. So as I start to get, or as we start to get, a buildup of this inside, what's going to happen with this is in here and not out here in this extracellular fluid? What's going to happen is 2 things. One, the inside of this is going to start to increase in pH. In other words, this is going to start to become more basic. That's going to start to become more basic. But because there's less of these out here now, if we take these away, this starts to become more acidic. The pH starts to drop, right? So we start to get a decrease in pH. Now remember, this is all over because I'm going to go and draw up here right now. So we're going to start to get a decrease in pH, right? So what's happened is my nerve is fired. I've got all these potassiums. The potassium is changing the polarity of this membrane, and then what's going to happen is we're going to get this bicarbonate coming in, and then we're going to get a decrease in pH. Now, when we get a decrease in pH, you have channels now that are on the axon. And what these do is normally these are going to allow sodium to come in. So here's my sodium once again, right? But it's also supposed to allow my bicarbonate to come in. Okay, and bicarbonate come in. And on top of that, normally what's going to happen is I'm going to get some chlorine going out, right? So now, when this pH changes and it becomes more acidic, this is closed off. So this doesn't work anymore. That's closed off. So now, if this isn't coming into the inside of the axon, what's going to happen to the inside of the axon if it doesn't have as much base coming in? The pH on the inside of the axon is also going to decrease. Remember, if we decrease the pH, it becomes more acidic. If we increase the pH, it becomes more basic. That's why I'm getting more base molecules. I'm increasing the pH. It's becoming more of a base. This is becoming more acidic. As the outside environment becomes more acidic, the inside environment becomes more acidic because this cannot enter into the neuron. When this becomes more acidic, then what happens now is the excitability of this neuron decreases. You're going to start to get a decrease in action potentials. Okay, so that's the role that's going to happen with astrocytes when it comes to maintaining homeostasis and how often this can fire also. Okay, so that's a lot for astrocytes. As you can see, they do a ton of stuff. So that's it. Thank you so much for watching. If you enjoyed the video, please hit the like and subscribe button, and we will catch you next time. Thank you again for watching.